Outcomes of patients with relapsed refractory Philadelphia chromosome-positive ALL are very poor, even in the modern TKI era. So, we have Ponatinib, which is a very highly potent TKI. But even in the relapsed refractory setting, you can get response rates of maybe 40%. But in the large PACE study, the 1-year progression-free survival was only 8%. So these are not durable remissions. So there's some preclinical evidence that Philadelphia chromosome-positive ALL is very dependent on BCL2 for survival, and so this raised the potential therapeutic role of venetoclax for this disease. And there are also some preclinical studies suggesting potential synergy between Ponatinib and venetoclax. And this was thought to potentially be mediated by the ability of Ponatinib to also target the Lin tyrosine kinase. And so inhibition of the Lin tyrosine kinase prevents upregulation of MCL1, which is also a known mechanism of resistance to venetoclax-based regimens. So together, this was the rationale for this particular combination in Philadelphia chromosome-positive ALL. We conducted a phase 1 study, a phase 1/2 study, and we're reporting the phase 1 results at ASH this year. Of this combination of Ponatinib, venetoclax, and dexamethasone for patients with relapsed refractory Philadelphia chromosome-positive ALL, or we did allow patients with CML and lymphoid blast phase. Patients had to have received at least one BCR-ABL TKI. And because of the potential toxicity of Ponatinib, patients could not have any uncontrolled cardiovascular disease. And also, because we were particularly interested in the role of venetoclax in this setting, they couldn't have had any prior venetoclax. So the way that the study was designed is we initially, in cycle 1, we gave patients 45 milligrams of Ponatinib daily. There was an initial 7-day lead-in of Ponatinib monotherapy. And then followed on day 8, the combination of venetoclax, which we did a ramp up to either 400 milligrams or 800 milligrams, depending on the dose level. And then also beginning on day 8, 4 days of dexamethasone 40 milligrams daily. And then in cycles 2 and beyond, the dose of Ponatinib was adjusted as we've done in other studies. So the Ponatinib dose either dropped to 30 milligrams for patients who had achieved a remission, or for patients who had achieved a complete molecular remission, which is absence of a detectable BCR-ABL by PCR. Those patients were decreased to 15 milligrams of Ponatinib. And then they continued on the same dose of venetoclax. It was given continuously, either 400 milligrams daily or 800 milligrams daily, and then 4 days of dexamethasone each cycle. We also give CNS prophylaxis to all patients, and then rituximab was added for patients who were CD20 positive. So we treated 9 patients in the phase 1 portion of the study. So, of course, for a phase 1 study, the main endpoint was to determine the maximum tolerated dose of venetoclax in the combination. So we treated 3 patients at the 400 milligram daily dose, and then 6 patients at the 800 milligram daily dose. We actually had no DLTs observed. 800 milligrams of venetoclax was the highest dose that we were looking to explore. So that is what we're considering the recommended phase 2 dose for future study. Overall, the adverse events are similar to what you would expect with this combination. There was some myelosuppression, particularly at the 800 milligram dose, but this was easily addressed, generally speaking, by interrupting the dose of venetoclax and in some patients reducing the dose. Even though this is we were talking about the phase 1 portion of the study, we of course wanted to look at response rates and durability of response. So we were very encouraged that the overall response rate, so the combination of CR and CRI, was 56%. So 5 out of the 9 patients, 4 5 also achieved a complete molecular response, and we saw these responses across different groups. So we saw, ranging in the 50-60% range of response in patients with prior Ponatinib or even prior blinatumomab or prior transplant. And then one particularly interesting thing that we saw was that although the numbers are small, of the 3 patients who treated with the 400 milligram daily dose of venetoclax, none responded. However, 5 of the 6 treated with the 800 milligram daily dose responded. So the response rate is 83%. in the group who received the venetoclax 800 milligrams daily. And of all the responses, with a median follow-up of nearly a year, none of them have relapsed. So that's very encouraging, potential durable responses that we're seeing. So this is a population that we really have very limited options. And as I said, we know Ponatinib is an effective drug, but I think the potential implications of what we're seeing in terms of what appear to be higher response rates, higher rates of deep response, as indicated by the complete molecular remission rates, and also some initial evidence of very durable responses, that this is an excellent alternative to Ponatinib single agent, certainly in the relapsed refractory setting. And importantly, this is an entirely oral regimen, oral and chemotherapy-free regimen. So this, is a very easy regimen to give to patients in the setting. And with a high complete molecular response rate, the goal is that these patients could potentially be bridged to transplant. And so I think this opens up the potential role of venetoclax in this disease in the relapsed refractory setting. And then if we see continued very encouraging data for durability responses, this is something that could also potentially be evaluated in the frontline setting and integrating venetoclax into regimens for the treatment of Philadelphia chromosome-positive ALL. I would again, emphasize that this is an entirely chemotherapy-free, entirely oral regimen. That we are seeing durable responses in. No patients have relapsed to date with a median follow-up of 10 to 12 months or so. And this compares very favorably to the historical data with Ponatinib alone, where the 1 year progression-free survival was only 8%. So I do think these responses are much more durable that we see with Ponatinib single agent. So it emphasizes the potential role of venetoclax in this disease.